Thank you for being here. My name is Gelabert, currently a postdoctoral researcher at the University of Vienna, working with Professor Pinhasi, and here I'm presenting you human and mammalian nuclear DNA and metagenomes from a 25,000 years old sediment from Satsurblia cave in Georgia. The images that I am using in this presentation are mostly from an unsubmitted publication, so I would kindly ask you not to share any of these images because we would like to keep it confidential. Before going deep into the methods and the results that I am going to present about this publication, I would like you to introduce into which is the current knowledge of the genomics of the Paleolithic in Europe, as well as which is, up to me, the most relevant publication that has preceded us in the study of ancient DNA from soil sediments. This here you can see a very nice image from Fu et al. 2016 publication, in which in the left you can see a heat map which represents the shared genetic diversity between pairs of upper Paleolithic genomes, and in the right you can see a multidimensional scaling built with the same individuals, which basically illustrates the presence of different genetic clusters in the upper Paleolithic in Europe. If you want to go back, Kostenki and Goyet, both individuals dated around 35,000 years old, are the oldest genomes in which we can trace a kind of genetic continuity between the present-day population and these genomes. As I have told you, there's this publication from Sloan et al. From 2017, which is a landmark in the field of paleogenomics from sediments from caves. Not only because the team was able to recover multiple mammalian empty genomes from different caves all across Europe, also because they were able to perform some phylogenetic analysis with hominin sequences from soil, which is a landmark in the field, and it's the first time that these kind of results are published. Our study was performed with samples from Satsurblia cave, which is a cave located in the southern Caucasus, close to other caves that have also been inhabited in the upper Paleolithic, such as Tudzwana cave. Satsurblia cave was firstly excavated in 1975 and has been excavated multiple times. Our samples, all of them, were taken in the campaign of 2016. Two different occupation areas have been discovered in Satsurblia cave, area A and area B. We studied both of them, taking samples from both areas and from different layers, but I am going only to focus on Satsurblia 29 sample, which is from area B and layer 3, because it gave an outstanding amount of human DNA presence. This sample is from this layer B3, which is very rich in archaeological material and has been radiocarbon dated with an estimated age between 25.5 and 24.4 thousand years old. So why this sample is incredible? This sample is very important because Satsurblia 29 showed an extreme amount of human presence, which was around 3%. So in order to study the sample, we sequenced 500 million sequencing grids, and then we classified them with a fast metagenomic approach with centrifuge, and we were able to detect the presence of possibly 4 different mammalian species. Then we built a composite sequence with these 4 mammalian genomes, and we align all the sequencing rings against these 4 mammalian reference genomes. And both the length distribution and also the determination pattern of the aligned rates against this reference indicate the clear signal of ancient DNA present on the sample, as the length distribution indicates that most of the reads are very short, and the deamination pattern is showing the presence of high levels of deamination in the read length ends, which is typical from ancient DNA. Then we took the reads that align to the human reference. In this case, were 600,000 reads. Then we filtered those reads, and finally we called we randomly genotype the 1.2 million positions from the array that has mostly been used to study ancient genomes. So we first built a principal component analysis with the whole Eurasian diversity available nowadays, and then we projected multiple ancient genomes. Here you can see Satsurblia 29 sample, which appears close to Botswana, to sample from the Botswana cave and from an age estimated around the same as Satsurblia 29. These two samples, which cluster together, are differentiated from the other genome from Satsurblia cave that has an estimated age of 15,000 years old, and Cotias cave with an estimated age around the same as Satsurblia from 15,000 years old. So this is indicating the presence of a cluster built by Satsurblia 29 and Botswana 2, which is separated from the rest of clusters of the genomes of the Upper Paleolithic in Europe. To deeply explore these connections, we use an F3 outgroup statistics analysis and also an admixture analysis, and both analyses indicate that Satsurblia 29 sample shows strong genetic affinities with Botswana 2 sample, indicating the presence of this connection and this new genetic lineage. We also took advantage of the empty DNA in order to explore the connections of Satsurblia 29 samples with the rest of available Mitochondrial from the Upper Paleolithic, and we saw that Satsurblia 29 first has haplogroup N and is closely placed together with Botswana 3 individual, also from the Botswana cave and from in a similar age to Botswana 2, and also with some samples from a cave in Bulgaria that have been dated to be 45,000 years old. So this is first indicating that the credibility of the sample from soil from Satsurblia 29 sample, and also indicating that this sample belongs to a very old lineage previous to the last glacial maximum in Europe. We also used this mitochondrial genome in order to perform a molecular dating with BEAST, with this Bayesian approach, and to see if the molecular dating was concordant with the radiocarbon dates that we have available from the soil layer. Most of the models that we tried gave us very old molecular dates. However, the confidence intervals of these datings are very broad, so most of them, in most of cases, the radiocarbon date is inside the confidence intervals, saying that the molecular dating is plausible and is concordant with the radiocarbon dates. So finally, just to sum it up, the different ideas and to give you some clues about what are we going to do next, I would like to remark the importance of this project. It is the first time which we are evidencing the capacity to recover genomic material from soil, not only to recover mitochondrial genomes, also to recover partial genomic sequences. And it's the first time that these genomic sequences from soil and from an Upper Paleolithic cave have been used to perform some genomic analysis, which is also a very important landmark in the field. Using this genomic material, we have been able to discover a new human genetic lineage that inhabited southern Caucasus before the last glacial maximum. And in the future, we are planning to extend this project, sampling much more samples from Satsurblia cave in order to try to find other human genetic sequences, and also tending to sequence more sequences from other mammalians that could give us information related to the interaction between human and other mammalian species, as well as other information regarding ecology or diversity at that time in the Caucasus. So thank you very much for your attention, and this would not have been able with the collaboration of multiple teams from different institutions. Thank you very much.